This video will cover subtopic D.4 The Heart. So here's a list of understandings, applications, and skills that will be covered in this video. So the majority of this video will deal with heart rhythms and the structures that control the rhythmic beating of the heart. So these diagrams are really serve as a reminder of the structures that do control that rhythmic beating of the heart. Recall that all heart contractions start in the sinoatrial node. That sinoatrial node is that specialized bundle of nerve cells that will initialize the wave of depolarization that spreads initially throughout the atrium and will lead to the atrium contracting. That signal that starts at the sinoatrial node will pass to the arterioventricular node. At the arterioventricular node, the signal does get delayed a little bit to allow the atrium to contract. Then the signal will travel from the arterioventricular node to the ventricles, allowing the ventricles to contract. So this video will cover more of the specifics of that signal traveling from the sinoatrial node to the arterioventricular node, how the signal gets delayed, as well as the specialized nerve fibers found in the septum of the heart, and the specialized nerve fibers found on the muscle linings just outside of the ventricles as well. So this rhythmic beating of the heart is going to be performed by cardiac muscle. So cardiac muscle is one of three types of muscles, which include skeletal muscle and smooth muscle. So the muscle is involuntary and striated in appearance. So meaning involuntary, meaning we don't actually control the beating of our heart. It happens without us thinking about it. And the muscle cells have these striations in them, these lines in them, when viewed under a microscope. And these striations are important for the contraction of the muscle, for the forceful contraction of the muscle cell. So in some ways, cardiac muscle appears very similar to skeletal muscle in terms of the striations that it contains. However, the shape of a cardiac muscle cell is going to be different. It's weird looking in a way. So the cells are Y-shaped, not like your typical round shape or elongated, like literally it forms like a Y. So in the diagram here, I'm just going to draw a line here because this is the end of one cell. And then what I've outlined here is one entire cell. So, it's not exactly a Y, but it does have a Y shape to it. So it has a branch point to it. And so the cells are going to be Y-shaped, joined at the end by a complex network of interconnected cells. So basically, it allows the cell can be connected to multiple cells. So each cell is connected to more than one cell. And then the spot where the cells connect to each other is generally referred to as an intercalated disc. So the intercalated discs are junctions that will connect one cell to another by a double membrane gap junction. Gap junctions are basically proteins that can connect two cell membranes together. So that intercalated disc is basically the round part of the cell here. This is where this one cell will connect with another cell. And then between these membranes, remember the membranes are going to be distinct membranes. They're not going to mesh together to form one membrane. But kind of connecting those membranes are these porous protein channels called gap junctions. And this will allow that electrical signal to pass from one cell directly into the next. So this interconnection through these gap junctions is what allows waves of depolarization to easily pass through each cell and pass along each cell, leading to a synchronization of muscle contractions. So when these cardiac muscle cells contract, they need to contract as one. So the gap junctions allow that to happen. And the fact that these cells are interconnected allows them to depolarize and contract in unison. So these diagrams just kind of show different perspectives of what those cardiac muscle cells would look like. So this diagram here shows a networking branch of cardiac muscle cells. So again, like here you have one cell, and you have these branch points, and then these lines are also those intercalated discs that contain those gap junctions. So as a wave of depolarization passes through one cell, it can easily pass from one cell to the next directly, so no need for like neurotransmitters or anything like that. And even as like the depolarization, passes from one cell, it can pass to multiple cells at the same time. So it allows for a rapid depolarization and a synchronized depolarization. The diagram here just shows how the cardiac muscle is organized relative to other types of muscle. So another diagram just showing the organization of that cardiac muscle cell. So we have a histological picture of cardiac muscle. So the dark purple stain is going to be those intercalated discs. The intercalated disc, again, is basically where one cell ends, and it's a disc because it's kind of flat and round, essentially. And the membrane will have these projections that can allow overlap with an adjacent membrane. And then there are these proteins found in the intercalated disc that will connect one membrane to the other. And this will allow that wave of depolarization to pass from one cell to another. So those muscle cells work together to create that cardiac cycle. The cardiac cycle is a repeating sequence of actions in the heart, which pumps blood throughout the body. So remember, there's the two parts, the diastole and the systole. So the diastole is a part of the cardiac cycle when the heart refills, following systole, which is the contraction. So atrium contraction, ventricular contraction, and then there is a brief moment where the heart refills with blood. So you have blood coming in from the vena cava into the atrium, which is also coming into the ventricle, and then you have blood coming in from the pulmonary vein into the into the ventricle, which is, or atrium, sorry, which is passing into the ventricle. So the diastole can be divided into two parts. There's the ventricular diastole, which is the period in which the ventricles are filling and relaxing, and the atrial diastole is a period during which the atria are relaxing. So as mentioned at the beginning of the video, this wave of depolarization that causes those cardiac muscles to contract, creating that cardiac cycle, is going to start at the sinoatrial node. So the sinoatrial node is the heart's natural pacemaker. The sinoatrial node consists of a cluster of cells that are situated in the upper part of the wall of the right atrium. So in the right atrium, you have that bundle of nerves, which is the sinoatrial node, which makes up the sinoatrial node. So it is here that electrical pulse impulses are generated. The gap junctions in the heart muscle cells will allow the electrical impulses to flow freely between the cells and spread rapidly across the atrium. So even though that wave of depolarization starts here, those gap junctions just allow that signal to spread all throughout the atrium. So the signal from the sinoatrial node will contract only the atrium, the right and left atrium. The signal from the sinoatrial node also passes to the arterioventricular node, which then spreads through the heart tissue called Purkinje fibers, contracting the ventricles. This contraction will cause the systole and the arterioventricular valves to shut. During diastole, the arterioventricular valves will open and the ventricles fill with blood. When the ventricles are about 70% filled, then the cycle will end. So this diagram just goes through the different pressure changes at different points in that cardiac cycle. So this diagram should look familiar from your test and from your textbook. And then now you're seeing it again in the option units. So, the different lines represent the different parts of the heart. So here, we have the yellow line, which represents the atrial pressure. So, when there is the contraction of the atrium, you get the arterioventricular valves are closing. And then at the end of this, the arterioventricular valves will open. The green line represents the ventricular pressure when the ventricles contract, and it contracts with a lot of force, so there is a lot of pressure as well. And then the red line represents the pressure experienced by the aorta, which at the height of the ventricle contraction, the aorta will be under similar pressure as well. So once that signal passes from the sinoatrial node to the arterioventricular node, there will be a delay of that signal. So the contraction of the atria and the ventricle is delayed by about 0.12 seconds. It's a very, very brief delay. The signal from the sinoatrial node will take time to travel to the arterioventricular node, and the arterioventricular node cells take longer to become excited. So they don't they take a bit of time to propagate that signal. The delay is due to a number of factors. So the arterioventricular nodes are smaller and slower because there are reduced sodium channels. That's part of the reason why they're slower. And they have a greater negative resting potential and a longer refractory period. There is also fewer gap junctions, which basically all leads to it being more non-conductive tissue, all kind of taken together. The delay in conduction will allow the atria to contract and empty the blood into the ventricle before the ventricles contract. So you need the atrium to contract first and then the ventricles to contract. So the delay in the signal at the arterioventricular node is what allows the atrium to contract and then the ventricles to contract. So a contraction too early would lead to too small a volume of blood entering the ventricles. So from the arterioventricular node, that electrical signal will pass to specialized structures called the Purkinje fibers and the bundle of His. So the bundle of His is a collection of heart muscle cells specialized for electrical conduction. They transmit the electrical impulses from the arterioventricular node to the point of the heart apex by the bundle branches. So from the arterioventricular node, there is a specialized structure called the bundle of His. And from the bundle of His, the signal will then pass down to these other structures referred to as the bundle branches. So that electrical signal will pass down those bundle branches located in that septum of the heart and will travel along these nerve fibers along the wall of the heart called Purkinje fibers. So the bundle branches will lead to those Purkinje fibers, which will provide electrical conduction to the ventricles, which will cause the cardiac muscle of the ventricles to contract. So it's a signal starting to travel up the Purkinje fibers that will cause the ventricle to contract. The high-speed conduction of the fibers will occur due to modifications of the fibers, including there's relatively few myofibrils, there's a big diameter of the cells, a higher density of sodium channels, and a high number of mitochondria, so it allows the signal to spread quickly. So the cardiac cycle can also be mapped using equipment or a technique called an electrocardiogram, or ECG. An ECG will measure the voltage given off by the sinoatrial node and the arterioventricular node of the heart. Now there is a characteristic shape to the cardiac cycle. I've just kind of drawn a line between the two waves to indicate where one cardiac cycle would be. So, like I said, there are characteristic shapes to an ECG of the heart, and each of these points will identify a particular point in the cardiac cycle. So there is this little wave at the beginning called the P wave. This is the voltage given off by the sinoatrial node, which indicates atrial systole. At point Q, you get a little bit of a dip. This is where the arterioventricular node sends its impulse, so where things kind of get slowed down a little bit, and where things start moving again. So then you get this QRX complex, you get this big spike in voltage, and basically this is where the impulses from the arterioventricular node spread down the conducting fibers and out to the Purkinje fibers, signaling the ventricular systole. And then you get the T wave, which is the arterioventricular node repolarizing. Now, the repolarization of the sinoatrial node is in here, but it's overshadowed by the large QRS complex. And basically, the whole process will then repeat itself for that cardiac cycle. So now, the cardiac cycle is something that is rhythmic. It is always occurring. And so sometimes things can go wrong. The rhythmic control of that cardiac cycle can be interrupted, or it can be too fast, or it can be too slow. And so sometimes interventions are needed to moderate that cardiac cycle. So this is where artificial pacemakers come into play. So a pacemaker is a small battery-operated device that senses when your heart is beating irregularly or too slowly. The pacemaker is implanted, well, at least the battery component is either implanted just below the chest, or you can probably have it above, but it's most likely implanted into the chest as well. There are electrical leads from that battery, which go into the heart itself, and then you have these electrical leads that touch various parts of the heart that will stimulate your heart to beat. So it sends a signal to your heart that makes your heart beat at the correct pace, so it makes a pace. So pacemakers may be used for people who have heart problems that cause their heart to beat too slowly. A slow heartbeat is called bradycardia. Some pacemakers can be used to stop a heart rate that is too fast or irregular, and that's referred to as tachycardia. So, if in the previous slide, you had that normal ECG trace of the heart, something like tachycardia, you'd have shorter interspersed QRS complexes occurring very rapidly. That would indicate a heart that is beating very quickly. So, as well, if you have ventricular fibrillation, this is an uncontrolled contraction of the ventricle. This is basically like a heart that is not pumping properly or beating properly, so little blood is going to be pumped. This is where a defibrillator can come in handy, which we'll talk about in a bit. So defibrillators, or defibrillation, is a process in which an electric device gives an electric shock to the heart. So that electric shock to the heart will help reestablish normal contraction rhythms in a heart having dangerous arrhythmia or in cardiac arrest. So in recent years, small portable defibrillators have become available. These are called automated external defibrillators, or AEDs, and are often found in commercial spaces or buildings. There's two of them here at White Oaks, one in the South Campus, one in the North Campus, and they're usually located by main entrances. See if you can find where they're located. So this diagram just shows how an AED could be operated. So there's usually diagrams indicating where the electrical lead should be placed, and then auditory instructions telling you when to push the button to send a charge. Now, other conditions that can affect that cardiac cycle include hypertension and thrombosis. So thrombosis, as we've talked about previously, is the formation of a blood clot inside a blood vessel, obstructing the flow of blood through the circulatory system. Thrombosis can occur due to arteriosclerosis, which is the hardening of the arteries due to the accumulation of plaque. That plaque will often develop because of high levels of lipids in the blood. The plaque will reduce the speed of blood flow, triggering a clot, which can completely obstruct blood flow. So if this is your normal healthy artery, a plaque buildup is basically a deposition of fat under the lining of the artery or the artery wall. And then that buildup of plaque will contain of lipids, will also contain cells in there, which are releasing clotting factors. So if that plaque were to rupture, you would have clotting factors that get released into the blood, which could trigger the formation of a blood clot. If that clot is big enough, it can completely obstruct the flow of blood through that blood vessel, leading to things like pulmonary embolism, a heart attack, or a stroke. So one of the common tools now to determine if a cardiac cycle is, or the heart is performing as it should, is the stethoscope. The stethoscope was invented by a person by the name of René Laennec in 1819. So it's 200 years now since the invention of the stethoscope. So when he made his creation, he used it to learn abnormal and normal breathing sounds, and from this, Laennec was able to develop the skills needed to diagnose diseases such as bronchitis, pneumonia, and tuberculosis. And as well, it can be used for listening to the heart, determining if it is beating normally. The invention of the stethoscope has led to vastly improved knowledge of the workings of the chest at the time. And the diagram here just shows that basic structure of a stethoscope. So there are those two parts that go into your ear, and then the portion that is placed onto the chest or the back to listen to someone's breathing or to listen to their heart beating. And then placing the stethoscope at particular positions can allow particular information about the heart to be heard or to be determined. So hypertension now is a slowing down and blocking of blood flow, and when this happens, it will put more pressure on the arteries, and this is what's called hypertension. So constant high blood pressure has many negative consequences, such as a high risk of stroke, kidney failure, or aneurysm. Blood pressure is basically the measure of the force of blood pushing outwards on the walls of the artery. The more blood your heart pumps, and the narrower the arteries are, the higher the blood pressure. And greater incidences of hypertension can occur for smokers, elderly, postmenopause females, people that are sedentary, people who with high salt and high fat diets. So with blood pressure now, when reading blood pressure, and this can be used with a stethoscope or a fancy blood pressure monitoring device, blood pressure readings will have two values to them. So there'll be the systolic pressure and the diastolic pressure. So the systolic pressure is going to be the top number, and then the diastolic pressure is the lower number. So the systolic pressure will measure the pressure in the arteries when the heart beats, and the diastolic pressure measures the pressure in the arteries when the heart muscle is resting. So you generally will get two numbers out of this. And so the way that blood pressure is measured is using a device called a sphygmomanometer. That's that cuff that will go around the arm. And this cuff will get inflated to a very high pressure. And then the doctor will be wearing a stethoscope with it placed on the arm just below the cuff. So once the cuff is inflated, blood flow in the artery is basically going to be stopped. And so what the doctor is listening for is for when that blood flow returns, or listening for when that blood flow comes back, and that will indicate the systolic pressure, and then they listen for when that sound disappears at that particular pressure, and then that will indicate the diastolic pressure. So that is the end of this video for D.4 The Heart.